My name is Sophany Phauk, and I'm original from Cambodia. And I'm going to present with the research title on the host parasites, the interactions and the bacterial communities of the parasitoid DRYINIDAE and HALICTOPHAGIDES on the important insect pest, CICADELLIDAE. So to give you introduction, so I'm going to give some background. So I think many people in participants already gave some of what is Auchenorrhyncha. Yes, I did that. But, there's more than 42 described species, or more than that now. I think some new species from these regions going to be described, I think. And as we already know that the living Leafhoppers in the superfamily Membranocoides and comprise of two subfamily, so CICADELLIDAE and Membranocoides, and most of them are significant agricultural pests. And at least 20,000 now was described, and those are to feed on the plant stuff, as you already know, and can be transmitted like in the morning presentation, so a virus. So for example, in my case, the Planthopper that the three of the CICADELLIDAE or a Leafhopper that I'm gonna talk, which is more focused on the bacteria that you can see in the couple. So this is somehow the whole morphologies that looks like here, and as you already know, like in Southeast Asia, we are mostly on crop productions, more likely into rice production, and you can see the two species of the green leafhopper, so GLH and other most common leafhoppers on dry crop, and they primarily critical because they spread the virus species such as a chancre virus. So this photo I got it from the Aries, and then you can see the symptoms. So they will spreading those kind of the virus, and then you can see the leaf of the plants will getting green, yellows. So this is the represent for the disease. And one importance that I just want to mention, because that really related to my study. So we knew that the Candida salses, which is provide essential amino acids, deficient in their plants diet, and they already and we already know that they have a long co-evolutionaries between the host and the bacteria. Okay, in general, so this is the project's objective for my research in overall. So I'm trying to investigate the ecological communities of the microorganisms associated with the superfamily, so that the members coidae, and with the insect species whom they interact with, so such as they can be parasitoids, they can be naturalistics, so they can live together, and they respond to the forest and agricultural ecosystem in Cambodia. So the photos represent for the naturalism. So you can see the weaver ant or the Cophila smelling gaga. So this one is the widespread species of ant in Southeast Asia, and you probably seen the membranes goes closely around those the ant. So they cannot more getting a benefit between each other. So the membranes so release the honeydews. So and then the ant will go and then collect those kind of the stuffs, and in return, so they will protect the hopper. And the other things, I think you already seen some of the a photo from Greece. So there are some of the parasitisms, which is you can see in the photo. So mostly the CICADELLIDAE, and then you can, if you look at the So if you look at this photo, you can see the a sacs, a larval sacs, or we call it a parasite. So this is the larval of the Auchenorrhyncha species, and they always hanging at the under the not the abdomen, the thorax of the hopper. So, okay, I'm going to go in deep into my research project for this presentation. So, I just want to use a diagram. So, we have host, and then we have a parasite, and then the parasite was parasitoids on the host, and then this is one objective that we be able to see that how they are interaction between the host and the parasites. And also we want to see that, so we will look at, so what is the complexity of the microbiomes in between the host and the parasite? So how they look like, what are the compositions, what is the communities between those kind of two or interaction species. And okay, so this you probably knew that the most famous and really got over a lot of work with the parasites and from Korea, and I hope that you knew that. And this is the story that telling that something with when people from outside and then they try to invade it to the wealthy family in those houses, and then they start to act as the host, which and then there are some of the environment changes between inside the house. So this is a represent what I'm going to infer to what they looks like between the interaction between the host and parasite. So there are some of the questions and also the objective what we are looking for. So finding the interactions between the host and CICADELLIDAE and also the parasites. And we want to see the composition so that we have the text inside those one, which is bacteria is one of the things that we are looking for. And then this is a similar study into the ecologies that Valerie is also mentioned. So we will use the alpha and beta diversity to see the inside or in the between from the sample to the sample how the bacterial community changes between the host and parasite insects. Okay, so I will be stick a little bit, but to just mention that I think in Southeast Asia we use a lot of pesticides also for controlling in terms of the agricultural practice. So to prevent all the pests, as well as we knew that the populations of those insects also are suppressed by the predator and parasites. So those parasites we knew that was found on leafhopper in the family of CICADELLIDAE, and they also play a really vital role in the biological control agents. And there are some of, I think in the US, I think I read in 1980 or something, I think probably Chris know that. So at least like more than 200 species of CICADELLIDAE was parasited by the DRYINIDAE and HALICTOPHAGIDAE. So yes, so that was the two group of the parasite have been reported interaction within the CICADELLIDAE. So this is some of the photos on the left one. So this is the CICADELLIDAE, and this is the host, and this is the male CICADELLIDAE, and this is the we call it inactive, not inactive. It's not movements. so they just live forever inside the host, and that was the female. And the right photo is represent for the Auchenorrhyncha wasp. So those one is parasitoids. So you can see that there's a small sac underneath the body, and this is the adult size. So and that you can see a really difference from what the two parasites. And one study also provided interactions, the networking between, to study the networking between the parasites and the host, how they are interaction from one species to another. Okay, I just want to brief to give you the biologist's point of view in terms of the life cycle. So for Strepsiptera, so they are the life cycle is the same as the whole metamorphosis or complete metamorphosis, but they are given into another term, which is the hypermetamorphosis, which is the same type, but they are more complicated. So if you look at the photo here, so this is when they are emerging. So they have a different part of the host. So the host, for the male one, you can see this, they living in a different host. Like for this one is the beaver ants. And then they emerge from the abdomen of the host. And then the host still alive. And then this is the adult. But comparing to the female one, so they have a different host, or maybe the same host, but they kind of more parasites female larvae, so they live forever in the host. And we can say that a neo-actinic female, which is they not both, they don't fly, just only for mating, waiting the male to come. And this is the Hymenoptera, so the Trinity mostly. So you can see that the way when the adult, so this is the female, and then they have a shelly, so like a hook that are be able to grab when for the prey. So this is the hopper new, and you can see that was like there's some of the sacs underneath their abdomens. And this is some of the life cycle. So both are complete with metamorphosis. So okay, so now I just want to introduce where I'm studying. So the site that I'm selecting was in Cambodia, and then mostly near the border, near around Tonle Sap, which is the biggest lake in Southeast Asia, I can say that, and freshwater. And the habitat type is look like similar in the Philippines. So we have buffalo, water buffalo, and mostly the gradient of the habitat is rice fields, along the rice field. And okay, so again, so this is larval sacs, and this is a HALICTOPHAGIDE larval. So the insect mostly dissect separately between the host and the parasites. So sampling and data processing is kind of more complicated, but I'm not going to go in deep to that. But to just let you know that we have 90 sample, and those one is separated into 3 for the 30 for the non-infects, 30 for the infects, and 30 was the parasitoid that dissected from the infects, insects. So all the sample was collected in Cambodia, and then we did the DNA extractions, which is because mostly we will, for the CO1, we want to identify, because the dissects parasitoid was not be able to, I mean all the sample was delivered from the wild, So we didn't use the rearing or feeding in the lab. So then for those one for the morphologies of the host will be transferred to Sweden. So in the Uppsala University and SLU, so Swedish University of Agriculture. And then we need to focus on the specific regions, so 16S and only the hyper regions of the V3 and V4 to look at the bacterial communities. And then all those will be with the high technology SciLife lab at the Uppsala University. And then we process all the data through the bioinformatic pipeline for the microbial communities. And those one is, we have a special pipeline for our team, which is focused on the bacteria. Okay, and this is some of the results that we got based on the CO1 genes. So you can see that because, as I mentioned, we cannot identify based on the larval. So we use CO1 as instead. And then, and also the database in the internet or from the previous study was not available. But we divide it into group. So there's a 5 group from the DRYINIDAE and 4 group from the SEARCA. Okay, so this is the result that host interactions. So we can, yes. But in summary, so we can see that at least 3 species of the host was parasitoids by the DRYINIDAE and 5 species parasitoids by the group of HALICTOPHAGIDAE. And this is really important. So we call it the amplicon sequence variants, which is a really short fragments of the DNA. So just only 400 to 450 base pair. And then we produce all the data into the pipeline, and then we identify 300 AS3. So that was a sequence represent for the bacteria. And then we identify almost 900,000 was sequenced through the data set. And this is somehow that related to, we can see that 33 AS3 sharing the bacteria, sharing between the host and parasites. And 69 was a similarity bacteria between the AS3 of the parasitoid DRYINIDAE and HALICTOPHAGIDAE. So this is the kind of the composition. So I'm going to show you based on the phylum and also the genus. And this one is the infect host, a normal host, and a parasite, a DRYINIDAE, and HALICTOPHAGIDAE. And the total, we can see that 11 bacteria phyla was found in the data set. And those the dominant bacteria was bacteria Odata, Proteobacteria, Firmicutes, and Actinobacteria, Biota. So bacterial compositions in specific only on the host. So we found only 5 bacteria phyla. And the most dominant one was Bacterial Odata, which is 70%. And followed by the Proteobacterial Odata, and they are the most dominant. For the parasitoids and DRYINIDAE, so we see that the Proteobacteria was the most dominant, which is 55% in the DRYINIDAE. But if you look at the parasitoid HALICTOPHAGIDAE, so there's 9 phyla, and the dominant one was also Proteobacteria, so 77%. Okay, so we want to see what this looks like in the between the host and also the parasitoid. So we used the alpha diversity, and we found that the highest richness, the bacterial communities, was on the Dryiinidae. And based on the observations of richness, Shannon and Simpson index. We also compared that the Kruskal Valley spread test indicate that there was strong significant difference between the host and parasites. And we also look at pairwise comparisons to see that how they are different or not. So they are different except observed between richness between Dryiinidae and HALICTOPHAGIDAE. So we also look at the lift across space from one group to another group. And this is somehow more complicated, but I'm not going to go deep with that. But I just want to give you some of the idea how the bacterial community look like between the host and parasites. So no highly distinct separately were observed based on the ordination, as you can see. And however, the Pilanova tests show significant difference of bacterial community across the insect family, also the insect family and host parasitoids. And the host parasite group, like this is a more deep, I'm gonna skip this one. You can ask me later if you want. And we're also comparing a non-host and infect host to see how they are different in terms of the bacteria's compositions or communities. And if you look at that, there's no clustering together by, we have 6 species of the host. But based on the ordination, they gave us a pattern with separate between the infects and the non-infect hosts. And this is also ego did that, and then we also apply for the bacterial community as well. So we call it indicator species analysis, which is we look at what kind of species that are specifically associated to the group. And then we have two categories. So we have a sample type, which is based on the host and the parasites. And the other one we based on the insects family. And so we have 3 insect family. And okay, so the most important, we found that 18 AS3 were associated in the sample type. And 31 indicator AS3 were associated in this group. And interestingly, so if you look at the host or the CICADELLIDAE, as Candidata salcium was the specific which means associated into the host and the CICADELLIDAE. But interestingly, at the parasitoid HALICTOPHAGIDAE, so Wolbachia was found the most associated into that group. So okay, so we found at least 6 there's a 9 parasitic group parasites on the 6 host species of the CICADELLIDAE. And this is some of the outcome by comparing the paper from Vietnam. So we found that the Auchenorrhyncha weird. So this trend can be confirmed as a new record for Cambodia. And we found that there are sharings between the host and parasites by bacteria, at least 33 HHV birds identified. Okay, and 5 bacteria phyla were found in the host, and Candidata salcius on those symbiont was the most dominant. And we already know that there's a strongly co-evolution between those kind of group and bacteria. Okay, and also confirmed by the ISA's salcia was a specific associated in that CICADELLIDAE. So, Yes, and one more thing is that we should know that bacterial community between the normal and infect host CICADELLIDAE show no difference, which means the bacteria was no effects on the host. So all related regarding to the bacterial communities. And yes, could be the first report or study host parasite associated and inside to their microbiota insects. So again, if you look at that, I might have a question to mind or use that we really get more information on that. For me, I still that have a question like one of the example like the Madridoscato. I think Dimitri probably knew very clearly. So there's one small inside the big, small inside the big one, and big one inside another big one. So it seems like we have more to learn in terms of the dipping inside into the bacterial communities. So there's still questions of that how we are going to work more. And yes. Okay, thank you. Oh, thank you for my team. So my supervisors, and that's all my team. And also the supporting project, which is supported by SIA, Swedish International Development Study, for my research. Thank you.